Hello, my name is Andreas. I'm a senior science fellow in Wuppertal in pharma, doing computational molecular design. And it's my pleasure to introduce Andreas Bender. We know each other for many years now. And actually, Andreas currently has too many jobs to name them all. He is founder of a company that also will introduce their product tomorrow and pitch that. And he is a lecturer in Cambridge, and he was working in the past in AstraZeneca and before in Novartis. And yeah, actually, Andreas started with chemistry, but also with his interest in programming. And from there, he got into data science, and his real interest is in making data make decisions and drive decisions. And that's what the talk is about, as you can see, from AI to decisions, from models to impact. And with that, I hand over to you.
Great. Yeah, thanks for the introduction. So we heard about AI, right? We've seen some fancy videos in the beginning. Everyone talks about that, even my grandmother. But the question is, if you do life science, right, and you want to make predictions and you want to drive decisions, how do you do that in practice, right? Which type of omics data generated how? So how do you put that into practice? That's the question I'm trying to address. And so the next 20 minutes or so, I will reflect a bit on those areas because that's where the difficulties creep in, right? Concepts are easy, putting it into practice is not so easy anymore. And that's what we are trying to do. So anything I say here is in my capacity as an academic. I usually say that so I can say what I want. If you want to look up some of the details of what I say, a few years ago, we wrote this article, AI and Drug Discovery, what is realistic, what are illusions? So that discusses the data that we have in the drug discovery area, which is very different from other areas. So we had images of insects, right? You can label them. Is that true in drug discovery data? Well, it's not. Things depend on dose and endotype and 20 other things, right? You cannot even label your data, right? And so on. And so that's what we discuss in this article. It's generally understandable. If you're a computer scientist, a biologist, look at this article. So what we'll talk about is the current state of AI in drug discovery, methods validation. We will talk about data and proxy assays. Do we actually have the right data to train our models? And I will give some examples of where I think data can really be used for decision making. And then there's, if I have time, a few slides on psychology, which often drives decisions as well. So where are we right now? That's where we were in 1981. That was the cover picture of Fortune Business Magazine, and they proclaimed the next industrial revolution. At Merck, actually German Merck, they refer to. That was structural modeling, right? SGI was founded that year. So docking software was published this year. I entered primary school. Was very graphically driven that year, that cycle of computer-aided drug discovery. The second one was around 2000, biotech bubble. Lion Biosciences was worth $1 billion, right? The human genome was sequenced, much more data-driven around 2000. And right now, that's what the headlines look like today. So that's 2018 to 2020, but they are still the same today if you read them. So how AI is changing drug discovery, which implies clinical success, right? Changing drug discovery doesn't mean changing the process, it means changing success. A few things though changed. A few things are similar, a few things changed. We have much more data, right? Think about single cell omics and so on. We have ontologies, bioassay ontology, of course not always implemented, but we can metadata annotations we have GPUs and so on. Many things changed. What did not change though is which preclinical data point predicts for the clinic, right? Efficacy and safety for the clinic. We can generate terabytes of data. I need 1 bit of data, right? Does that compound succeed in the clinic? 1 bit, not terabytes, right? Of course, the question is which bit, but yeah. So a few things changed, but this crucial element didn't really, in my opinion. It's easy to overestimate things. That's a few headlines from just after the human genome sequence was published. Human genome was published. There were expectations we will have 3,000 and 10,000 drug targets. That was the expectation in 2001. 16 years later, there was a review that put that number at 667. So not 3,000 or 10,000, right? So not all the expectations come true. So the question is, we can generate data, we can do lots of stuff, but is it tech push or science pull, right? And tech push is one thing, and it's great. You can try new things, nothing wrong, but you also need science pull. Yeah, we need to do what we should do, not what we can do. That's the point here. Okay, so lots of funding went into AI and drug discovery, whatever the precise numbers are, it's lots of that, went down a little bit in 22 and 23. What did we get for that? So on the left-hand side, you see AI first drug discovery companies. This data from a NRDD article in 2022. And you see, if you have AI first drug discovery companies, you see lots of projects in green, so discovery and preclinical, okay? So what does that mean? Lots of preclinical modeling, bioactivity modeling, and so on, very in vitro-based modeling, if you wish, right? That's what many companies do, because it's not about tackling the translational aspect, right? It's modeling bioactivity data, binding, and so on. But progression to the clinic, basically you don't see much of. There's currently only, I think, one compound in phase 3. It's the recursion compound, but it's for quite a niche indication. So you see many of the projects in those companies are still in discovery or preclinical. And big pharma looks different. So that's on the right. You see more balanced portfolio. There was more contraction to certain disease areas, probably, so numbers go down, mergers, terminated projects. But you see you have many more projects in late stages, right? So basically, the proof point is still outstanding. Of course, there was the Excientia compound, the benevolent compound, and so on that failed in phase 3. So that's the only one left that I'm aware of. That's a bit of a fun fact update. So you remember the headlines from 2018 to 2020, right? How AI is changing drug discovery. This is from an article much later, and people are rolling back a bit. So inside the nascent industry of AI-designed drugs, there's no shortcuts to drug discovery. These trials are still in their early days, yeah. So it's quite funny how language changes if you read that. So anyway, conclusion, lots of activity, right? Clinical proof point is still outstanding. If you look into chemistry, targets, there's also not as much novelty as you expect, because you need to model your data. Where do you have data? Well, on GPCRs, kinases, whatever you explored before, right? And so there's less novelty than you would expect if you look into that in detail. Always think about in terms of input and output. What you want to care about is output, right? Clinical success. Input is 100 million of funding, great, but you need to do something with it, right? And then the output is actually what matters, in my opinion, in the end. What I came across recently was how Paul Janssen actually did drug discovery. I can very much recommend that. I mean, he discovered 48 or so drugs in 40 years, and he had a very much person-based approach, not process, person-based approach. I can really recommend that article. Okay, let's talk about validation and data. So in theory, and we've seen that before, when classifying insect images, we have a method, we have labeled data, we can see how well the method works. In drug discovery, that is actually quite difficult to do. Why? Because many of the labels that we have, they're actually more in vitro relevant, right? You have log-D values and so on. You can model them quite well, actually. But the question is, what does that mean for in vivo, right? Everything else is just a proxy, right? How does that translate? Validation is costly, prospective validation, and chemical space is huge, right? So how do you actually validate? How is your sample size large enough? How is it unbiased? It's basically infeasible in practical terms, right? So how do you actually validate? That's a big problem. But one way, of course, to see that is drug discovery is actually proof by example, right? That's what some of my friends told me as well, right? It's not actually about proving that an algorithm works. After a while, you gain experience. Maybe that's the only way you can really validate an algorithm. Scientifically, not very fulfilling, I have to say. But still, what else can you do? I didn't come up with much better yet when it comes to in vivo relevance, translation, large numbers in a diverse space, right? Unbiased space. How do you do that in practice? And there's one reason of that, and that is chemical space. So this is, if you train a model, every galaxy is a scaffold, is a compound cluster or a series of compounds. You have a training set, your validation set, your external test set. Yeah, often you split it, right? So overlaps a bit, okay. So, but your next project set, right? Where's that? It's somewhere else, right? New scaffold series, right? So every model that you have, bioactivity model, is essentially a local model in many cases, except where your descriptors or method can extrapolate, right? Log-D, maybe some Fiscam properties and so on, but very difficult to do with bioactivity in practice, yeah? And that's one of the, it's very different actually in protein design space, right? Because you have evolution, right? So you have a much lower dimensionality of your data. In chemical space, you don't have that, yeah? You have 10 to the 60 compounds, you don't understand how they relate. There's also the way we think about models and use them. I think there's problems with that. So this is, if you're in academia and you want to publish in Nature Machine Learning, that's what you need to consider. So you have a model, you put in your data, you go to good AUC, and you get your paper. That's all you need to care about. But of course, reality is a bit more complex because you need to put that model output in practice, right? So what's the follow-up assay to run in practice, right? There's also, on the input side, of course, every project is different. Do you work on lifestyle disease or terminal cancer, right? What's your CMX? What's your dose in men, right? Does your model that is based on a 10 micromolar cutoff, does that actually apply to that PK environment and so on, right? In many cases, the answer is, well, not really. So the question is, if you have those models based on in vitro data that don't correspond to the in vivo relevant environment, be it cell line, time point concentration, and so on, right? What does the model output mean in the disease context, in vivo context, not in vitro context, right? So there's a disconnect between the two. And basically the point is that if you have a model, right, that's one thing, but there's a process you want to improve, right? And you have interfaces, and that's what matters in practice. I'll skip that one. We heard about explainable AI before. It's a great thing. It's problematic in chemical space though. So there's one paper we published just after my PhD. It's my own paper, so I can bash that. And we try to identify features of bitter compounds, okay? Compounds which you can take, they taste bitter. So what are those features? If you're a chemist, you look at those structures, what do you see? I mean, those features, they are basically sugar rings, right? And that is, what does that illustrate? Biases and confounding factors in databases. If you have a natural product, it's quite often bitter. If you have a natural product, it's quite often glycosylated. You have sugar rings, okay? If you have other data sets, for example, you have chemical series, right? You have synthesis bias. You make analogs of a compound in a project, yeah? And if you want to apply explainable AI methods to those data sets, quite often you find something you know already, right? And it's very difficult to go away from that bias in practice in chemical space, incredibly difficult. It might work in other areas. In chemical space, I think it's very, very difficult. So let's talk about data. So which data matters? So here we modeled, there's also from the article I mentioned before, what actually happens across the phases if you're faster, 20% faster, if you have 20% lower failure rates in relative terms, and if you have reduced the cost per phase by 20%. And you see that the highest bars you have in the clinical phases. So if you increase the chance of success in relative terms by 20%, right? It's all time and cost and failure rates. It's all mapped on a monetary axis on the left, so you can compare them. And what you see is in the early stages, sure, cost reduction helps a little bit, but what really matters is the clinical phases. Why is that? You have high failure rates. They take long and they are costly, right? So phase 2 efficacy is basically what you need to care about. But that's not where much of the AI methods, right, that we have in preclinical discovery. They care about the preclinical part, and that's good. You can gain something. There's nothing wrong with that. But it's like looking for the key where the light is, right? I mean, the key might be there, yeah, but it might also be somewhere else. And I think in this case, the key is basically when it comes to clinical translation. And you see that in cancer, if you have cancer biomarkers, you go from 3 to 8% success rates, for example, right? If you can do endotyping, you see the impact of that, and that directly relates to chances of success in clinical phase 2. Yeah, you have two types of data, right? One is more in vitro data, and that's great. You can label it, but how does that translate to in vivo, right? And that's kind of the two different types, if you speak very broadly, that you can have in the life sciences. And I will come to the point then why classifying images does not necessarily translate to drug discovery. I will come to that in a second. So that's how I thought until actually 2008. Andreas mentioned I was at Novartis, I was a postdoc, and I was dealing with high content screening data. And I'm a chemist. Yeah. So, and as a chemist, if you for the first time look at high content screening data, I had difficulty sleeping for half a year because I didn't understand that data. After a while, I realized that's how biology is. Yeah. And then I came to appreciate that. But the point is, you cannot just label your data in many cases. So, in a simple worldview, right, a compound is toxic. Well, depends on dose. Compound binds to a target. Well, depends on distribution, right? Does it get into a target tissue? A compound treats the disease. Well, depends on endotype, right? We had that in asthma, diabetes, I mean, you need to endotype your diseases. So the simple labels basically don't really work. And that's a problem, right? Because these would be in vivo relevant labels, but we don't really have them. We cannot properly endotype in many cases. Yeah. And that conditionality of data is actually across the board. if you think about pharmacology, right, which type of downstream signaling do we have? If you think about diseases, whether you have ICD-10 or whatever classification you want, right, it's symptom clusters, CNS diseases, right? I mean, it's not ICD-10 codes and distinct diseases, not at all. So we cannot label that either, and so on. And things depend on those and so on, right? So it's difficult to label the data that we have, especially in the in vivo relevant area. One example, adverse reactions. Sometimes you see machine learning publications that try to answer that question. Does it cause a certain drug, cause an adverse drug reaction? Well, if you look into that, right, that's what your data looks like. So what's actually the genotype? Then you have a certain disease endotype. What's the dose of that drug? How do you dose? Maybe only in males with that subisoform after 3 months of dosing, you see a certain side effect in 12% of cases, and some are severe, some are not, and so on. So that's what your real world data looks like, right? So does a certain drug cause an adverse event? Well, difficult to say. Even water can be toxic, right? If you drink 3 liters of water in half an hour, changes your electrolyte levels, even that can be toxic. So would that be a toxic compound? Probably not, right? So labeling data is difficult. I think you got the point probably by now. So why is that a problem for drug discovery, that we have so much data generated with proxy assays right now? So why do we have so many proxy assays actually? So high throughput screening came up, right, in the 80s, late 80s. And then the question was, so I have 10,000 hits. So what do I do with that? So I need to profile further, right? And then there are other simple assays, let's say BSEP inhibition or so, if you want to approximate one mechanism of DILI and so on. And you profile compounds, not you specifically, but we as an industry, in a myriad of different ways. But those assays represent only one part of the in vivo translation, right? One part of the mechanisms that we have. But that means we have quite a lot of proxy assay data right now that get repurposed in generative models, for example, to predict compounds that work in vivo. But that limitations of the data often get forgotten. The limitations of the data and the PK aspects. Yeah? What does that mean at dose in men, actually? Yeah. And there's a second disconnect basically between the model and the environment. One was not caring about the interfaces. What's the follow-up assay? What's my disease context? But also the types of data we have that we can actually put into models. Yeah, so they're quite detached from each other, and that's of course a problem in practice. That's what we need to work on. And the domains, they work quite differently. So here's the image domain, the chemical domain, and the biological domain. We've seen image classification approaches before, but in those areas you have an object that you can represent as a matrix. Then you have your CNN, for example, convolutional network that detects eyes, so to speak, in the later layers, certain shapes and so on. And then your output layer tells you that's a cat, for example. So you have labeled data. It's largely unconditional, right? In the chemical domain, your compound can be reactive, can have different conformations, different tautomeric states, different polymorphs. It can have, I mean, like many things can change about that compound, right? There's not one representation of the cat, or in this case, the compound. Goes back to one of the previous questions. What's the right representation? Well, we don't know in many cases. And so we can calculate many descriptors. We can use many different models. And then our labels are conditional as well, right? And that's the big difference. In the first case, we have a matrix. CNN operates on matrix. Labels unconditional, great. Okay? But in the other cases, biology is similar to chemistry, even worse in a way. That's actually very different, right? So here, green, integration of object in the machine learning pipeline, but that's very different in the other cases, right? Yeah, and this is why another reason, in addition to the conditionality of the data, why machine learning in our area is actually much more difficult to implement. Yeah, a few final slides here. So where do I believe that machine learning actually does help? So one area is in vivo PK prediction. I mean, for 10 years or so, we now live in times of pharma on in vivo red measurements, I guess. So we have quite homogeneous data of a few thousand compounds. And actually people from Bayer, Sebastian Schneckner and Andreas, actually pioneered that, I might say, I think 2017 or so with those papers. We at Astra did that as well. And that, I think, is basically models that translate to in vivo, right? It's in vivo relevant data. And that's something I quite like. They are not predictive across the whole range in many cases, at least when we validate it, but you can remove compounds from DMTA cycles with very high clearance, for example, right? And that's in some cases even good enough. Yeah, you don't need to rank everything necessarily. You want to remove what you don't want. Yeah, you can use models like that as well, or at least at Astra in those times, that's how they were used. Another area where we use data is gene expression-based compound selection. So we did that to differentiate cells, for example, cancer cells, de-differentiate cancer cells, and so on, push them into apoptosis. And I think right now there's actually, that's one example where we differentiated stem cells to cardiomyocytes, and it's after. Oh yeah, no worries, in a minute I'm finished. It differentiates stem cells into cardiomyocytes, and that's probably the closest I felt to being got to see those cells beating under the microscope, yeah? Because the computer selected chemicals, you put them on cells, 3 weeks later, they beat under the microscope. And I think right now, there's kind of a revival of that as well. So we work with a Swedish company, Aska Therapeutics, to differentiate cancer cells in vivo, so that they're representing antigens, cancer antigens. So you can do CAR T cell therapy, right? You take out your your T cells, and then you engineer the CAR into the cell, so they attack the cancer cells with the antigen. But what you can do as well is you have your cancer cell, you have a small molecule, much easier PK-wise and so on, administration-wise, and you induce the cells into dendritic cells, so they represent the antigens as well. So that's the idea here. So we kind of revive that right now. Started a few companies. One of them, Pangea Bio, will present tomorrow, actually. Yeah, that's, I think, if I still have 1 minute, there's a few aspects that actually drive decisions, right? And how the world operates that are not necessarily scientific and technical. So it's a bit light here, but it's still quite serious in a way. So AI is where it is also because of human psychology, right? So what happens there sometimes, there's lots of fear of missing out. It's usually, if your 16-year-old teenager wants to go to a party, right? And you say, nah, stay at home. Yeah, then of course there's fear of missing out. They could have kissed Jimmy that day or something like that, right? Also happens on the management level in big companies sometimes, right? From that emerges the ideal startup strategy equation. Take hot air from a startup. So don't tell anyone what you do, but generate hot air, right? And if that then it combines with FOMO from Big Pharma, right? There's a perception of secret sauce that can happen quite easily as well. And that happens on multiple levels, individual psychology as well as organizational psychology, right? Everyone needs a winner. So if someone gives you 20 million, right, to implement the new AI strategy, and your boss asks you after 2 years, so what happens with those 20 million? Yeah, what was successful? It's unlikely you come back and say, well, actually nothing, right? So everyone needs a winner, yeah, to represent that in a good light. And there's quite a lot of selective reporting of successes. And that makes it difficult to compare algorithms, right? Everything seems to work, but that's not quite true, right? I mean, it cannot be true, right? Otherwise, clinical success rates would be high. So it's difficult to advance the field if you cannot really compare methods very well. Okay, so we need to analyze our data, absolutely. AI is a valuable tool in the toolbox. The real game changer will come once we can translate, right? Understanding which early stage data is really predictive for in vivo. So consortia for data sharing. Also data generation, actually, using experimental design. There were, of course, quite a lot of AMI consortia and so on. But the problem is, if you do not design data in the way you need them later, you don't cover chemical space, you don't have the metadata and so on. Yeah. So that doesn't always work out in the long run. Okay, so methods need to translate to reality. So we need to go from model validation to process validation. Thanks for listening.